Hello, everyone. Welcome, everybody to the virtual ILC Symposium. I think as many of you know, we had planned this last year as an in-person event, but obviously couldn't do it. And after a lot of discussion, decided to have this as a virtual symposium. Just very quickly to introduce myself, I'm Steffi Oesterreich. I'm a professor here at the University of Pittsburgh at UPMC Hillman Cancer Center. I'm very happy to co-chair this event with Drs. Christine Desmedt, who will also deliver the keynote presentation in a few minutes. Dr. Desmedt is head of the Laboratory of Translational Breast Cancer Research at KU Leuven, Belgium. The other two co-chairs are Dr. Otto Metzger, medical oncologist at Dana-Farber Cancer Institute in Boston and executive officer at for the Alliance for Clinical Trials in Oncology at Harvard Medical School. And the fourth co-chair is Dr. Patrick Dirksen, a professor at the University of Medical Center in Utrecht in the Netherlands. We are very happy that you join us here, and I'm very happy to report that we have a total at this moment 656 registered attendees from 35 countries, which is fantastic. And before I give it over to Christine, I just want to really thank everyone. I want to thank the organizing committee again, Christine, Patrick, Otto, fantastic. I want to thank a lot of people in the background who have helped, especially Nadine, who worked with Excel events to make this possible. Many others who have helped to organize this. And I think I will quickly again introduce Dr. Desmedt for the keynote lecture. Dr. Desmedt is head of the Laboratory for Translational Breast Cancer Research at KU Leuven in Belgium since 2018. She is co-chair of the ELBCC, and we will hear more about this with Dr. Patrick Dirksen and Salomon. And she's also co-chair of the Lobster Pot with Patrick Dirksen. And Christine, lucky for all of us and many on the call, on the meeting today, has been really highly engaged in lobular breast cancer research for many years. So if you have questions, please give it into the Q&A. Type the question there in the chat box. I want to remind all the speakers here on the backstage to mute yourself if you are not speaking. Otherwise, we get a strange background noise. Again, welcome everyone to this first virtual ILC symposium. And Dr. Desmedt, I'll give it over to you.
Thank you very much, Steffi, for the introduction. So, thank you very much, everyone, and welcome to this symposium. So it is my pleasure now in the coming 1 hour to take some time with you to review what we have learned about lobular breast cancer in the last years and how this creates opportunities for further individualizing treatments for these patients. So the outline of my presentation is the following. First, I'd like to say a word about the initiatives that are present now for joining forces for accelerating and improving research on lobular breast cancer. After that, I want to talk a bit about the importance of a correct, reproducible, and homogeneous diagnosis of ILC. After that, I'll try to give an overview about the use of multigene assays for patients with lobular breast cancer. After that, I will speak about the characterization of the tumor immune microenvironment. And then I will speak about the genomic characterization of primary lobular breast cancer and what has been done and what still remains to be done to advance research in the metastatic setting. So let's start with the first point. So I think that really I can say that we are very proud that these two consortiums, these two initiatives do exist. So on the left, you see the ELBCC, the European Lobular Breast Cancer Consortium, which was initially created by Patrick Dirksen and myself and Anne are now also co-chairs with Patrick. This really created already a very nice and engaged group at the European level to focus on important research questions and in order to work together at the European level. But this consortium is not only composed of scientists and clinicians, but also from highly engaged patient advocates, which can be organizations or individuals, as you might have heard yesterday. But the European side is really partnering with the US. And in this context, we're actually making the bonds tighter and tighter with the US and with the Lobular Breast Cancer Alliance, which is directed by Laurie Hutchison, which was at the scientific advisory board first led by Steffi and now by Otto Metzger. And the founding advocates and all the other advocates also play a fantastic role there, really to help this growing, to get the information to the patients, and also to get the scientists and the clinicians together. In the context of ELBCC, we were lucky to have actually a European cost action network funded. And this helped us to have some glue, some money, to have all these people together and to start some important work. And here I just want to highlight the working groups that we have in the context of lobster pots, which are the following. So the first working group is really working on guidelines for harmonized pathological and imaging diagnosis of lobular breast cancer. The second one aims to have an important prospective ILC collection, biobanking, data storage, and sharing. The third one focuses on omics approaches to define and stratify lobular breast cancer. The fourth one is involved in getting more experimental models because this is strongly needed to better investigate and model this disease. And finally, the last one is focused on ILC-specific clinical trial design. But all of this requires also an important communication at different levels with patients, within ourselves, and with the rest of the community. And this is why we also put that here in this network. So I'd just like to say also that what we are about to start here for the harmonized collection of sample and data was initially planned to be across Europe. But we have actually looked at expanding that also to non-European hospitals, and this will be possible. So we will soon have one large master protocol that can then be applied to large hospitals and institutions that see quite a lot of number of patients with lobular breast cancer, so that we can collect many more data than what has been collected so far in other large initiatives. Because we also want to investigate other type of data, like data related, for example, to adiposity, lifestyle, diet, physical activity. We also want to be able to collect not only tumor sample, but also normal mammary sample, metastatic samples, and plasma at different time points. And we want to do that also by collecting all different type of data in electronic case report forms, also by having central pathology here, and by this collection, by increasing the number of experimental models that we could have here for this disease. In terms of communication, I also want to do a little promotion here, since Patrick, Anne, and myself are actually editors of this special issue in Cancers on lobular breast cancer. On the right, you see the first articles that are already published, but there is still room to accept other and to review other articles. And the deadline for submission is the 31st of October. It has been extended as compared to the first deadline. And importantly, there is also a very interesting paper that was published very recently, which is really about stating how researchers, clinicians, and patient advocates can accelerate lobular breast cancer research. More will be said about that tomorrow in a dedicated session. And finally, in terms of communication, so we also have been working together with patient advocates to develop a leaflet that will be distributed both in paper but also on the website in different languages, so that newly diagnosed patients or their families could have the important information that they could have here. And I think for Europe, this is important because unlike US, where everyone understands and reads English, this is not the case for Europe. So the conclusion for the first part is that actually joining forces for accelerating and improving research in ILC really starts to become a reality. And there's a large collaboration, not only within the US, within Europe, but also it starts reaching other continents. So joining forces between patient advocates, clinicians, and researchers, I invite you to really join the panel discussion tomorrow. So now with regard to the second point, which is regarding towards a correct, reproducible, and homogeneous diagnosis of ILC. So why is a harmonized pathological diagnosis needed? Recently, there has been a central pathology review that has been done in 2 large clinical trials, the MINDAC trial and the West German study group Plan B trial. And here, out of the samples that were initially designated as lobular breast cancer samples, only 60 or 66% were recognized at the central pathology level to be really lobular patients. So local pathology really results mostly in overdiagnosis of ILC. This might be also very critical when retrospective studies are done on ended clinical trials where no central pathology has been done. So this could introduce some noise. And this is also important actually for the inclusion of lobular patients in dedicated trials. Additionally, and this will be further referred to by Dr. Matthias Christgen in the next session, but the reproducibility in the diagnosis is also very important. And he will report about his study where he assessed the reproducibility in diagnosis in 2 different case series, considering only H&E or H&E and E-cadherin immunohistochemistry. And he will tell you actually that the inter-observer agreement and also the agreement with the reference clearly is higher if immunohistochemistry results are considered. Finally, because of this evidence and what we have observed, what we want to do, and this is a work that has been initiated in the context of LobsterPot and ELBCC, we wanted to carry out a worldwide survey on how lobular breast cancer is diagnosed at the pathological level. And so far, we have been able to collect the answers from more than 150 pathologists across 36 countries, as you can see actually in the next slide. So you have here one little red dots represent the different countries that are participating. We started in Europe initially, but very rapidly expanded that actually to the different continents. So to one of these questions, there are many questions that we have asked, and I thank the pathologists in the room who may have answered this survey. Actually, we can see that globally half of the pathologists use actually immunohistochemistry and other do not. So, but it's not only that, there are many, many important findings that actually arise from this survey, like really the variety, for example, of E-cadherin antibodies that are used, some of which might not always be appropriate. So to conclude the second part, we can say that there is a discordance between local and central pathology for ILC diagnosis, and this should be acknowledged. Central pathology is important when performing retrospective analysis of large clinical trials and also when we want to include patients in ILC-specific trials. The survey that I was describing and that was currently done, I can still remain open for 1 or 2 weeks if you want. So if your pathologist, you still want to join, please do not hesitate to contact me. Also, we need to establish a homogeneous criteria to have this central pathology, pathology diagnosis of ILC. And finally, I want to say to stay tuned and also for the next session when you will hear much more about this. So now let's move to the third point, which is about multi-gene assays for patients with lobular breast cancer. So actually, several multi-gene prognostic assays have been developed, of which many are now commercially available. And you might know them, like the recurrence score, MammaPrint, Prosigna, EndoPredict, EpiClean, Breast Cancer Index, and MapOne. So the aim actually was to identify patients for whom the use of chemotherapy could be avoided to circumvent the overtreatment of patients with a low risk of disease recurrence. However, most of these assays have been actually developed independently of the histology of the disease. And so far, most of the investigation that has been done for patients with lobular is actually retrospective on larger series. And in this presentation, I now tried to gather and to put together all the evidence that we have so far. And apologies if I may have missed or might not give all the details because of time of all the different publications. So let's start with recurrence score. So here you see the different studies which have reported actually the evaluation of the recurrence score in lobular tumors. Here you see a table according to the original cut points for defining patients with low risk of recurrence, intermediate, and high risk. And as you can see here, you see that globally, the patients at high risk are very few according to this original cut point, and they range across around 2 to 3%. However, in a in a more recent study by Tadros and colleague, where they investigated and they compared classic lobular tumors to non-classic, they actually saw that patients with non-classic lobular tumor had a higher percentage of patients that had a higher risk of recurrence. When looking at the same recurrence score, but considering other cut points like the ones that have been used now in the TAILOR-X trial and the REXPONDER trial, we can see that here, as expected, there are a higher number of patients in the high risk category. And again, here we can see that also there are higher proportion in the non-classic ILC. But if we consider the population together, the general population of ILC, here the high risk is consistently around 8%. So what about the association between recurrence score and survival? So here I draw a table of two of the trials that were investigated that. The publication of Keysi et al. In 2017 was actually retrospectively looking at the SEER data, and therefore they had many, many patients. And here, actually, in terms of treatment, 20% received chemo. Median follow-up was not specified, and there was no central pathology done. The risk distribution was 21% low, 71% intermediate risk of relapse, and 8% high. When we look at the recent study from Matthias Christgen, there all patients from this trial actually received chemotherapy, and there were fewer patients here since they came from this clinical trial, 272 patients. Median follow-up was 5 years. There was a central pathology done, which, as I said before, really reduced the number of initially considered lobular tumors. And then when we look at the conclusions, on the left, we can see that patients with high risk recurrence score were associated with an increased rate of mortality. And also they showed that there was actually no benefit from adding chemotherapy in high and intermediate risk patients. In the publication from Christgen and colleague, actually here they showed that there was limited or no prognostic value of Oncotype DX for patients with ILC. However, we should consider that here there was a very small number of high-risk patients, 17, if I'm not wrong. So here, these results might be needed to be also further confirmed. And in this publication, they further emphasized that histological grade and lymph node status will still remain important prognostic parameters. When we look at MammaPrint, actually, we see that in a first retrospective analysis of different clinical trials, 24% in the publication of Boehmer et al. Were identified as high risk. In the recent presentation by Otto Metzger at EBCC, it showed that actually 16% of the patients of the lobular patients in the MINDAC trial after central pathology review were at high risk of recurrence. And when a distinction was done between classic ILC and the non-classic ILC, again, as before, we see here that there's an increased proportion of patients at high risk of recurrence in those that have non-classic ILC. So what about survival? So here, actually, this is the results shown by Otto Metzger during his presentation at EBCC. And what he showed here is actually, as you can see in the Kaplan-Meier curve on the left. So the plain lines are the lobular patients, the dotted lines are the ductal. So you can see that there is no real difference, and that's what they report, between patients that are at low risk of recurrence, being either ductal or lobular, and those that are at high risk of recurrence. Then another assay that has been investigated is EndoPredict with the clinical parameters. And this is the only one that is not only a multi-gene assay, but also considers actually tumor size and nodal status to have the risk identification. And these results were published last year also by Ivana Sestak in Clinical Cancer Research. And they showed actually that the 10-year risk of distant recurrence in the EP-clean low-risk groups was similar between lobular and ductal patients. And they claim that EP-clean is actually informative also in women with lobular breast cancer. So here actually you see an overview table that I made that compared actually the most recent studies that were presented or published. So only the first 4 actually reflect commercial tests, while the last one was added because this is actually a signature that was developed specifically for patients with lobular tumors. But I will not give more details now, since this will be also explained actually in the presentation later today. So what is important also to realize when we want to compare actually all the different percentages in the risk distribution and so on, is that all these different cohorts were not necessarily similar. Most of them come from clinical trials, but some combine different clinical trials. Also, if you look in terms of treatments, there can be a big difference. For example, for EP-clean that was investigated in the ABCSG 6, 8, and the ATAC trial, we see that here the patients received only endocrine treatment and no chemotherapy, and also that they were only postmenopausal patients. The median follow-up was relatively long. In MammaPrint, actually here, it was considering also the younger patients. And so this is a big difference actually from the other study. Also here you can see that there are differences in terms of patient characteristics, mainly in terms of age, and then also in terms of treatment that you see above. And you can see here again, actually, the main conclusions that have been derived by the authors based on their study. However, I want to tell you that just taking here this row by just comparing the proportion of patients with high risk of recurrence might be a bit too naive, because we need to take all these other parameters into consideration. So to conclude this part, I want to say that these tests have been evaluated in different patient cohorts, but this precludes a formal comparison because these are different patient populations that receive different treatments, have different characteristics, different durations of follow-up, and we know that this is important for patients with lobular tumors that may recur even later after the initial diagnosis. Not all studies considered central pathology for histological characterization. The difference in the proportion of patients assigned to the high-risk groups there is this strong difference actually according to the test that is used. But it seems that the histological subtyping of ILC is important because both for the recurrence score as for MammaPrint, we can see that there are more patients that are at high risk if they have non-classic ILC. Still, I think that special attention should be required for identifying late recurrences here, specifically for patients with ILC, and more work is needed to understand the use of these classifiers for patients with ILC. So to know more about this and also about the LOB6 signature, I also refer you to the presentation of Dr. Peter Simpson later today. Oops. So now, with regard to the characterization of the tumor immune microenvironment. So actually, to characterize the tumor microenvironment, I think that TIL assessment for tumor infiltrating lymphocyte has become quite standard now. So here, the focus is mainly to evaluate stromal TILs, because these have been shown to be also more reproducible between pathologists. And here you only evaluate the TILs which are located in the connective tissue within the tumor, in the connective tissue, as opposed to the intratumoral TILs. So what do we know so far in breast cancer? A short summary to see then after that the comparison with lobular tumors. So we know first, and I refer here to the figures and the tables that have been published by Carsten Denkert and the colleagues in Lancet Oncology in 2018. So we know that higher TIL levels are observed actually in triple negative breast cancer and HER2 positive breast cancer. But Carsten Denkert and colleagues have shown that higher TIL levels were associated with increased rates of pathological complete response in all molecular subtypes following preoperative chemotherapy treatment. So also in the luminal HER2 negative subgroup, as you can see here. What do we still also know in breast cancer is that higher TIL levels in patients with triple negative and HER2 positive breast cancer are associated with a better prognosis for DFS and OS, but that actually higher TIL levels are associated with worse prognosis in patients with ER positive, HER2 negative breast cancer. And this could be due to the relative resistance to adjuvant and metastatic endocrine treatment in these patients. And this actually is in line with two papers, one from Dembier et al. In Clinical Cancer Research in 2013, and another one by Anurag and colleague in JNCI in 2018. And these authors have globally shown that there are higher levels of immune infiltrates in luminal B as compared to luminal A tumors, but importantly, that higher levels of immune infiltrates are associated with reduced antiproliferative response to aromatase inhibition on one hand, and also with worse prognosis in these patients on the other hand. So the question is, what about these observations in lobular patients? So what do we know with regard to immune infiltrates in primary lobular tumors? So this is a work that we published 2-3 years ago. And here we can see on the left, actually, that the median levels of TILs in all lobular tumors, all the 600, a bit more than the 600 that we centrally assessed, was actually 5%, a median of 5% of TILs, of stromal TILs that were observed in these lobular tumors, although you can see that there was a very big spread here. And we could also see that when we were separating those tumors that were estrogen receptor positive, HER2 negative, from those that were triple negative or HER2 positive, that there were higher levels that were observed in these triple negative or HER2 positive tumors. When we did compare within another trial, an homogeneous series, the TIL levels between ER positive, HER2 negative IDC versus ILC, we saw significantly lower level actually in ILC as compared to IDC. So what about the associations of TILs with the standard clinical and pathological variables? So we see that in lobular tumors, we have more TILs in younger patients. in tumors where there's a high Ki67, so that are highly proliferative, and also in the mixed non-classic lobular tumors. However, when we compared here in the Big 298 trial, the associations in lobular tumors and in ductal tumors, we can see that the association with age could not be seen in patients with ductal tumors, but only in those with lobular tumors. Then with the collaboration of Emad Reka and the group in Nottingham, we were further looking at the expression at the immunohistochemistry level of different markers at the intratumoral, adjacent stromal, and distant stromal location between ER positive, HER2 negative ductal and lobular tumors. The p-values that you see here are at the univariate level here, and in parentheses at the multivariate level after correction for the standard clinical and pathological characteristics. And globally, you can see that actually lower levels of CD3 lymphocytes are observed actually in lobular tumors. And also we see lower levels of intratumoral cytotoxic T lymphocytes that we see here for the in the lobular tumors. We also see here lower levels of macrophages, actually in all different localizations. And we also see lower levels of FOXP3 positive cells, suggesting that it's not only in TILs, but that we see also different levels, different lower levels of the different immune subpopulations. We further investigated actually also the association between TILs and genomic alterations, and we found that actually tumors that had p53 ARID1A mutations were associated with higher TIL levels, while tumors with RBP3 mutations or HER3 mutations with lower levels. And in the same way, we found some copy number alterations that were associated with increased TIL levels. So what about TILs and prognosis in patients with ILC? Here I want to refer to the study that was conducted by TIL and colleagues, so conducted under the supervision of Anne Salomon at the Institut Curie. Here they carefully reviewed a bit more than 450 lobular tumors that were diagnosed at the Institut Curie between 2005 and 2008. And they globally found the same associations with the clinical and the clinical parameters as we reported before. But here of interest, when they considered these three subgroups of TIL negative, TIL below or equal to 5%, or TIL infiltration above 5%, they could see that this was actually associated with survival, and that higher TIL levels here were associated with a worse survival. And they further confirmed that here in a multivariate analysis, where they also took the other variables like the lymph node status and the molecular subtype into consideration. So what do we know then about TIL levels in a metastatic setting? This is something that we evaluated also together with colleagues recently. So actually, we see that we have lower TIL levels in primary tumors from patients who relapsed compared to published consecutive primary ILC cohort. And also we see lower TIL levels in primary, in the metastases as compared to the primary, although here this was not statistically significant. We also investigated the distribution of TIL levels according to the different organs where the metastases were present. And we actually see very big differences. However, here we cannot really have one strong conclusion, since per organ or per localization, there are too few samples actually to make a very strong and statistically relevant conclusion. The question is then, based on what we know here, is there a role for checkpoint inhibition actually to treat patients with lobular tumors? And this is actually the question that Dr. Marleen Kok is investigating in the Gelato trial together with her colleagues. And recently, her PhD student, Leonie Voorwerk, reported the first results at the ESMO Breast Cancer Conference this year. And she actually showed that the clinical benefits that could be seen by this treatment in these patients that had metastatic lobular breast tumor might be actually more concentrated in patients with triple negative lobular tumors as compared to those with ER positive disease. So to conclude this part, so we see that, and in general for breast cancer, we know that TIL levels are lower in ER positive, HER2 negative breast cancer as compared to triple negative and HER2 positive breast cancer. We know that high pre-treatment TIL levels are associated with increased PgR rates after neoadjuvant chemo in all breast cancer subtypes, but no specific data here are available for patients with lobular tumor. We also know that high pre-treatment TIL levels in ER-positive, HER2-negative breast cancer are associated with decreased anti-proliferative response to preoperative aromatase inhibition and with worse prognosis. But here again, there are so far no specific data for patients with ILC. So with regard to ILC, we know that the TIL levels are lower in ER-positive, HER2-negative ILC versus IDC. We also know that if there are more than 5% TILs present there, that this is associated with worse prognosis in these patients. We also have shown that the immune composition can be different in lobular versus ductal tumors. We also have seen that there are generally low levels of TILs actually in metastasis from ILC patients, and that these are even lower as compared to the primary tumor. And really more research is needed to understand organ-specific TIL levels and the benefit from immunotherapy. However, the tumor microenvironment is not only about TILs. Many other cells have an important role, like fibroblasts, adipocytes, and epithelial cells. And I invite you also to hear more about that in the talk tomorrow by Dr. Val Brunton. So now about the genomic characterization of primary lobular tumors. So several publications have been reporting actually the genomic landscape of lobular tumors, primary lobular tumors. And what I want to do now is just to highlight some of these features. So the first thing that you will notice is that actually in these three plots, the first line, so the most common genomic alteration is actually CDH1, which is the gene that codes for E-cadherin. And this is also what you see that is really the largest difference actually between lobular tumors and ductal tumors. So the alterations actually for in CDH1 are actually spread out throughout all the gene, as you can see here on the lollipop plot on the upper part of the figure. And in addition to these mutations, which are approximately found in 65% of all lobular tumors, we can also see a loss of one copy of the gene, so LOH, in approximately more than 90% of lobular tumors. And this you can clearly see the fact that most of these lobular tumors actually lost one copy of 16q. So why is this important, and why is this important in terms of personalizing treatment for patients with lobular tumors? This is because of the context of synthetic lethality. So synthetic lethality actually arises when a combination of deficiencies in the expression of two or more genes leads to cell death, whereas a deficiency in only one of these genes does not. And this is really what was actually looked at by the group of Chris Lord, and they found actually that inhibition of ROS1 actually in these tumors that also lack CDH1 could lead to synthetic lethality. And this is now what is being investigated in two clinical trials, the Rosaline trial in the early, in patients with early lobular tumors, and in the HOLO trial, actually in patients with metastatic E-cadherin negative breast cancer. Then something else that was really striking and that has been since then really reported consistently is actually the increased mutation rate of HER2 and also of HER3, but to a lesser extent mutations in patients with lobular tumors as compared to ductal tumors. We also previously reported actually that these mutations were also leading to an increased HER2 signature score, and that actually also patients that have HER2 mutations were associated with an increased risk of early relapse. So now HER2 mutations were also shown to be associated to endocrine resistance. And important trials have been developed to investigate the role for patients that are actually harboring activating HER2 mutations in their tumor, to combine actually to treat these patients with neratinib and to add also fulvestrant. So I will not speak too much about these data because I invite you actually to listen carefully to the talk of Dr. Ron Boos a bit later today to hear more about that. But this is really an area that we should be moving on to personalized treatment for patients with lobular breast cancer. Another important observation actually that was made in when studying these primary lobular tumors is that actually 40% of these tumors actually had an alteration that was converging to AKT activation. And this should also be taken together actually with findings by the group of Patrick Derksen that showed also that actually there was a strong PI3K AKT activation in lobular tumors, but they showed actually that this activation was independent of PIK3CA AKT-OP10 mutation. But here again, I will not tell you more about that, and I invite you to listen carefully also to the talk tomorrow of Patrick Derksen to explain you more about this. But here again, I think that this is really an important avenue to be further explored for personalizing treatment. So to conclude what we know, and here I was just putting it all together about the differences in the genomic landscape for primary lobular tumors, is that we know that CDH1 mutation LOH is really the hallmark of lobular tumors. And this is a therapeutic opportunity that is currently being explored. We also saw important differences in genomic alterations between ER-positive, HER2-negative ILC versus IDC. And many of these mutations actually are relevant because either they might be therapeutic targets like the HER2 mutations, but also they represent some markers of endocrine resistance. Also, in terms of survival, it has been shown that some mutations or copy number alterations are associated with worse survival, and also that patients with AKT1 or HER2 mutations have been associated with an increased risk of early relapse. So now to move on to the last part of my presentation, I want to talk a bit more about advancing research in metastatic lobular breast cancer. So far, when we look specifically first at the genomic research, which has been done in the context of metastatic breast cancer, actually, the research has been driven by samples that have been collected either in the context of clinical trials, and here I refer to the publication of Bertucci et al., either to institutional initiatives, and here, for example, to the beautiful and gigantic initiative by the MSK Impact. Also to patient partner research, which has been led by and is still led by Nikki Wagel, the Metastatic Breast Cancer Project, and also by international screening programs with Aurora as an example. So I think that without going into more details in all of these studies, what I want to tell you is that the most important message also for us here in the context of lobular tumors is that when comparing genomics between primary and metastatic tissues, actually most of these changes and differences are actually seen in hormonal receptor positive HER2 negative breast cancer, which actually encompasses 95% of all lobular tumors. And this was shown in the context of the MSK impact data, as you see here, with the levels, the fraction of samples that harbor these specific mutations in the primary sample, here in light green, or in the metastases. But be careful here, this is an unmatched comparison, meaning that these are global frequencies, but not pertaining to the same patient. Also, in the work by Bertucci et al. And the French colleagues, they reported that nearly all of the differences that were seen between early breast cancer and metastatic breast cancer were concentrated in those patients with hormonal receptor positive HER2 negative breast cancer. Then let's look at those studies that focused really on the investigation and the characterization of metastatic lobular tumors. Here we have the work of Sokol et al. That was also nicely driven by Steffi Oesterreich. Here they looked back at samples that were characterized by genomic health using targeted sequencing. Below you can see actually the origin of the metastatic samples that were analyzed here. And as expected, you can see that some localizations actually were more present in metastases, in metastatic sample from lobular as compared to ductal tumors. And this is consistent with the specific pattern of metastases that can be observed in these patients, such as an increased representation of gastrointestinal metastases or metastases in female reproductive organs. So, as to summarize, what they have seen is that actually they found in ILC as compared to IDC more frequently CDH1 alterations, NF1 mutations, TBX3 mutations, and PIK3CA mutations, while in IDC they more frequently saw MYC alterations, GATA3 mutations, FGFR1 amplifications, and TP53 alterations. We also, some time ago, then investigated the question of ESR1, because ESR1 is one of the most commonly known and investigated now source of endocrine resistance, with ESR1 mutations related to the resistance to aromatase inhibitors. And here, to do so, we investigated ESR1 mutations in primary and metastatic samples from 80 patients for which we had these matched primary and metastatic samples. We investigated the most commonly described and reported mutations using digital PCR. And actually, we reported first that there were no ESR1 mutation found both in the primary or the metastatic sample together from the same patient. However, and this is in a way a bit reassuring, we did not see any difference in type or prevalence of ESR1 mutation in metastatic ILC or IDC patients. And we did so by looking back at the MSK impact series and also in collaboration with Nick Turner in the SOPHIA and Paloma 3 trial. Using the same cohort, we further applied targeted sequencing to investigate the genomic landscape in these patients with matched primary and metastatic tumors. And to make a story short, we did find actually mutations or copy number alterations that were associated with endocrine resistance and that were private in the metastases in nearly 1 out of 4 or 1 out of 5 patients. And these mutations are like mutations, for example, in AKT1, ARID1A, ESR1, RBB2, and F1, or in terms of copy number alterations in P10, NF1, or CYP19A1. And this means that not all the information which is clinically relevant actually can be derived from the primary tumor. What I also want to say is that when looking at all these patients and comparing that, as I said also yesterday in my talk, is that every patient is unique and so is her cancer. Because here we draw this figure actually for a few patients in our publication, but every story, every disease, every treatment characteristics is different according to the patient. So and also the treatment schedule are very different, but we do get important information, and we really saw that globally, really in 50% of the patients, we can find actually alterations that are associated with endocrine resistance here. However, in all these studies that I've been reported, we were mainly working with metastatic biopsies, and this might be limiting since it might not represent the full metastasis, and it might not represent actually the full disease that is present in the body of a patient. So in this context, to learn more about disease progression and to learn more about treatment resistance, which can often be very heterogeneous even within, say, a single patient, post-mortem tissue donation programs actually offer the opportunity to have a complete resection or a multi-region sampling of each single lesion. They also have the opportunity to allow the resection of lesions that are otherwise not reachable. They also allow extensive liquid biopsies. So far, liquid biopsies have mainly been limited to blood samples, but here it gives us the opportunity to investigate also cerebrospinal fluid, ascites, pericardial fluid, pleural fluid, urine, saliva. It also allows us to really seek some histological confirmation for dubious aspects on imaging and to learn from that for the next patients, especially also in the context of lobular patients. And finally, it also will teach us what about incidental findings that were not seen on the imaging. However, even there are so many opportunities, the challenges are really quite large for this tissue donation program. Patient inclusion is not always easy, as it involves a very long and important discussion with the patient and with their family as well. And we want always to refer here to post-mortem tissue donation, to insist on donation, because we insist on the fact that this is really a patient-partnered study. The patient decides to donate tissue for further specific research here. But also the challenges involve logistics, because it requires a multidisciplinary expert team, extensive sample annotation and processing, transport and collaborations. And also it is important to keep post-mortem intervals short and to have a cold-controlled environment. And finally, which is also extremely important, is the high cost of these programs. Nevertheless, we took the challenge and we started last year in UZK Leuven the UPTIDER program, which is the UZK Leuven program for post-mortem tissue donation to enhance research. And we have multiple aims here, as you can see in this figure. So first, we want to have a complete histopathological characterization of all the lesions that are present within the patient. We also want to be able to very carefully reconstruct the disease evolution using phylogenetics. We want to understand treatment resistance, and I think here we have we have extremely good opportunities, because there are many drugs that are relatively recently now used for patients, like CDK4/6 inhibitors, neratinib, checkpoint inhibitors, for which we also see actually heterogeneous responses within the patient. And so here we have the ability to investigate why some lesions did respond and others not. We also aim to better characterize the tumor microenvironment, the metabolomics, to identify potentially new treatment targets, to also do some radiomic studies to further confront the findings with what we will have at the molecular level. We also want to investigate liquid biopsies, how representative the different sources, the different types of liquid biopsies are of the different metastases. We also want to generate new experimental models and to tackle disease heterogeneity. So to do so, we actually have a very large collaboration within UZ and KU Leuven, with many clinical departments and laboratories. So of course, we have a very close and excellent collaboration with the morgue and the forensic department. Also with the radiology department, because we, whenever possible, we do a post-mortem MRI. Also with the pathology department, with all the people in my lab that are all involved in that and very motivated for this project. Also with the multidisciplinary breast cancer team that is also extremely important and committed really to identify patients, to do the first important discussions with them, and also after that to be involved in the interpretation of the clinical data. But also the team involved in palliative care is extremely important to ensure a good communication with us, and then also with the biobank. But in addition to that, we also have a lot of collaborators actually within KU Leuven, within the VIB, and also externally. So to do the autopsy itself, we actually are generally a team of 10 to 12 persons, and the autopsy can last between 6 to 12 hours. We collect in general between 300 to 600 samples per autopsy, and every person has a specific role. I will not go too much into detail, but generally we work with 2 abductors, one person with a sterilizator to ensure that everything always is very sterile, also from one lesion to another. We have 1 coordinator to ensure that everything goes well according to the pre-established plan. We have different annotators that actually annotate the samples, make sure that the right labels are there, that the right conditions are recorded. Also, we have a photographer that take a piece, all the pictures from the larger samples and the samples after that before they are put in the different recipients. And then we have several people involved in sample processing. So also importantly, given the large number of clinical and sample data that we collect, we work with an electronic CRF that is being created in REDCap, and that is GDPR compliant. And we also use actually Lab Collector to record all the important information with regard to the sample and sample processing. So here I just want to show you one example, and to finish with that, from one patient with lobular tumors that was included in our study, and for which we performed the sample collection. So this is a patient that was diagnosed actually a bit more than 4 years ago with lobular tumors, lobular tumor that was estrogen receptor positive, progesterone positive, HER2 positive, and that was actually already presenting metastasis at diagnosis. And after several lines of endocrine chemotherapy or HER2-targeted therapy, the patient unfortunately died. So here I want actually to show an example from this patient here, from some lesions that we were taking. In total, we were identifying 57 actually metastatic lesions, of which 14 were incidental, so were not seen initially on the imaging. We also collected 7 different types of liquids. So actually here, what we see is actually a mass that was identified in the neck. And as you can see here, we then cut in different mirrored samples to be preserved according to different conditions. And this is actually here the hematoxylin eosin image that you see from the FFPE sample. So when you look here at the region that is identified by the red square, this is actually a region that after that we characterized using multiplex immunofluorescence, using the Milan technology here at the KU Leuven. So above you see the H&E, and below actually you see the staining here with the first round for DAPI, that is coloring the nuclei, and in red actually the estrogen receptor. What you see actually is that in this mass there were clearly 2 nodes that could not really be distinguished just looking at the H&E. But then when doing the immunofluorescence, you can see that actually the lymph node on the left was strongly positive for the estrogen receptor, as was the initial primary tumor, but not the lymph node which was immediately adjacent to it, that was only weakly positive or even negative for the estrogen receptor. So actually, the heterogeneity in estrogen receptor, progesterone receptor, and even HER2 expression is also something that we saw when we performed RNA sequencing on nearly 40 of these metastases. And this is as represented here for all the little dots that actually represent a different sample here. In this next image, what I want to show you is the intersample transcriptomic distances. So meaning that actually here, when it's dark blue, it means that the samples are extremely similar to each other. And the dark blue diagonal is just because it's the same sample. It's the distance between the same sample that you see on the Y and X axis. However, you can see that these distances are quite variable. And according to the clustering, you can see that what we see here below is mainly composed by actually the different samples of the primary tumor, which was still in place at the time of autopsy, together with some axillary lymph node, but also from some more distant lymph nodes in the body, while the other samples actually were mainly present here in the other side, but with a lot of actually transcriptomic heterogeneity between them. As another example, I also want to show you the copy number profiles actually of different sources of DNA that was present in different fluids. So here, for example, you can see the copy number profile of the cell-free DNA from the pleural fluid left on the left side. And you really can see that as expected, and it's really matching actually the profile of the primary tumor. You find here 16q loss, which is really consistent with the fact that it's a lobular tumor. You also do find HER2 amplification, as it was the case for this patient, and you find many more other alterations. But also actually in urine, where we did not expect to find a lot, we still found some little signal that we will be further investigating. And also in the ascites, so in the supernatant after centrifugation of ascites, we found a lot of representation of ctDNA actually. So to conclude this last part, we can say that actually, based on our first study, more than 50% of ILC metastases harbor genomic alterations that have previously been shown to be associated with endocrine resistance, that the matched primary metastasis comparison in our series revealed mutations and copy number alterations that were associated with endocrine resistance and that were only detected in the metastasis of the patients in 22 and 90%. And also that actually when comparing metastasis from ISC to metastasis from IDC metastasis, we could find an increase in CDH1, RBB2, FOXA1, and TBX3 mutations, and deletions and CDH1 deletions. And finally, as shown in the last slide, post-mortem tissue donation programs represent a unique opportunity to gather important insights in disease progression and treatment resistance. So overall, to conclude this presentation, I want to say that a correct diagnosis at the pathology level is crucial and that efforts are ongoing. The use of prognostic tests of this multi-gene test should be clarified for patients with ILC. That patient partner research on ILC is growing, that collaborative efforts across the continents are further being developed, that trials focusing on ILC are slowly showing up, but we really can do better, and that insights into disease progression and treatment resistance will hopefully allow to improve disease management of patients with ILC. So I want to finish here by showing you a slide that was made also by my PhD student, Karen van Balen, to show actually the different categories of trials ongoing for patients with ILC that focus on ER signaling pathway, synthetic lethality, CDH1, HER2 and HER2 mutations, or immune infiltrates. But we still need much more. So this is a picture of the consortium meeting from ELBCC in 2019. Hopefully we will be able to do another one soon in presence after all this COVID. And I want to thank everyone from my lab, from UZ Leuven, KU Leuven, from the large UPTIDER team, from my previous collaborators and colleagues at the Institut Jules Bordet, from collaborators at the University of Milan with Elia Biganzoli, with my collaborators at the NKI, University of Freiburg, Luxembourg, Institut Curie, Geneva, Cambridge, Nottingham, and all the other centers. And then also thanking for all the funding opportunities that make this research possible. And importantly, thanking all the patients and the family that are participating to these trials and to these studies to increase knowledge in lobular breast cancer. So thank you for your attention, and thank you for being patient to listen to me for 1 hour. Thank you.
Thank you very much, Dr. Desmedt. That was absolutely fantastic. Really a great start for the meeting. Very beautiful keynote lecture. Thank you. I think in the interest of time, we might move on. And, you could actually answer some of the questions in the Q&A chat box. Christine, that would be great. So today we will have 2 sessions. The first one on addressing challenges in lobular pathology, diagnosis and imaging, and the second one shared later on by Dr. Otto Metzger on addressing challenges in treatment of ILC. I will moderate the first session, and actually before we start this, I actually realized that I forgot to mention earlier the supporters and sponsors of the symposium. I really quickly want to do this. The main sponsor of the symposium is BCRF, the Breast Cancer Research Foundation, and the supporters are LBCA, Susan G. Komen, Lobular Breast Cancer UK, Lobular Island, Dynamy, and the ELBCC. So thanks again, everybody, for supporting this. So without further ado, I think I will start with introducing the first speaker for this session, which is Dr. Matthias Christgen. He's professor at the Medizinische Hochschule in Hannover. And Professor Christgen has worked on lobular for many years and really made some very fundamental discoveries and contribution to ILC research. And we are very happy that he joins us here today. The title of his presentation is Lobular Breast Cancer, Histomorphology and Different Concepts of a Special Spectrum of Tumors. Professor Christgen, please.
Hello. Very much for the kind introduction. I was asked to say a few words about the histomorphology of lobular breast cancer. I'm really happy to do so. For me as a pathologist, histomorphology is the most direct approach to the nature of cancer. Today, I would like to draw your attention to slightly different concepts of lobular in research and diagnostics. Our group in Hannover serves as a reference pathology for. So for instance, the Plan B trial and the WSG ADAPT trial, which has been a good success on this year's ASCO Congress, are supported by a central tissue banking, central pathology review, and biomarker assessment. And in this role as a reference pathology, we receive FFPE tissue tumor blocks from approximately different breast cancer centers from all over Germany. Now, this is a mammary carcinoma classified as ILC, invasive lobular breast cancer, by a local pathology in Germany. Our blinded central pathology review and confirmed the diagnosis of lobular breast cancer. However, breast cancer subtyping is not always consistent, and large clinical trials have recently shown the consistency of breast cancer subtyping in real-life diagnostics. In the WSG Plan B trial, which you can see on the left side of the slide, the central pathology ILC in approximately 66% of the cases classified as ILC by local assessment. Interestingly, similar data were reported from the MIND-ACT trial. In the MIND-ACT trial, Central Pathology Review confirmed the diagnosis of ILC in approximately 60% of the cases classified as ILC by local assessment. So ILC is slightly overdiagnosed in current practice, and this is represented here by the yellow pie chart pie pieces. And this finding encouraged us to reconsider the diagnostic concepts of ILC in research and diagnostics. My key message for today is that the concepts of ILC are slightly changing over time. Now, this is a schematic presentation showing in a Venn diagram-like manner between invasive lobular breast cancer, ILC on the one side, and mammary carcinoma, NST, on the other side. Please let me explain this in some. ILC was first described in the 1940s by Footy and Stewart, and they described ILC as a kind of breast cancer in mammary lobules that are affected by carcinoma in situ, ELCs. The diagnostic criteria for ILC include presence of a lobular carcinoma in situ and an invasive phase of loose isolated tumor cells coming from this lobular carcinoma in situ. And an advantage of this approach was the straightforward separation between ILC and non-lobular breast cancer based on the presence or absence of a lobular carcinoma in situ. And this is illustrated here in the schematic presentation by the strict vertical line between ILC and NST. In the 1970s, pathologists like Fechner and Azzopardi correctly pointed out that some otherwise typical ILCs simply lack an adjacent ELCs. Sometimes the ELCs was evident in the contralateral mammary gland. These studies rendered the diagnosis of ILC independent from the presence of. The proportion of ILCs associated with ELCs decreased from nearly 100% in some studies published in the 60s to about 50% in studies published in more recent years. And since the 1970s, the main diagnostic criteria for ILC were small loose tumor cells and growth in so-called single files, a term that was not coined by Footy and Stewart, but it was actually promoted and Azzopardi. And this definition of ILC is pretty much in line with the nowadays definition of ILC according to the World Health Organization. According to the WHO, ILC is defined as breast cancer composed of discohesive tumor cells individually dispersed or arranged in single files. And that means that ILC is essentially defined by the growth pattern and nothing else but the growth pattern. But the growth pattern of breast cancer is a wide morphological continuum, and there's a substantial proportion of tumors that show an indefinite growth pattern. I'm talking here about all the morphologically specific ILC variants, which can be exemptions from the rule. I'm just talking about the regular ILC in NSTs, and there's a substantial proportion of indefinite cases between these two entities on a morphological sense. Many, but not all, prefer to call these tumors mixed breast cancer NST/ILC. Mixed breast cancer NST/ILC accounts for approximately 2 to up to 18% of the breast cancers in published series. And the huge variation in the frequency of mixed breast cancer NST/ILC, also in its relation to the number of ILCs reported in the respective studies, illustrates the difficulties associated with defining a boundary between ILC and NST. In other words, the definition of ILC by the WHO is a simple and flexible and very respected definition. It's a good definition because it makes everyday's diagnostics easy for the pathologist. But I have to admit, in my opinion, and the drawback is that the boundary between ILC and NST is comparatively poorly defined. Now, loss of the cell adhesion molecule E-cadherin in ILC was first reported in 1993, and mutation of the corresponding gene, CDH1, was reported in 1995. Looking at published studies devoted specifically to ILC, you will find that the number of ILC cases reported to be E-cadherin negative or CDH1 mutant has been rising between the years 2000 and 2010. Now, granted, this may simply reflect improved DNA sequencing techniques, but this may also reflect an increased use of upfront immunohistochemical staining for E-cadherin to support the initial breast cancer subtyping. I believe ILC is increasingly perceived as a morphomolecular tumor entity defined by both combined morphological and molecular features related to impaired cell adhesion. Well, I have to admit I'm glad that I do not have to talk here about a very difficult topic, namely about defining all the genetic and/or epigenetic alterations that may impair cell adhesion and can induce a lobular tumor phenotype. This may also include other genes like alpha-catenin. But all I want to say from a conceptual point of view is that by considering ILC as a morphomolecular tumor entity, the term mixed breast cancer NST/ILC becomes problematic because it refers only to the morphology of the tumor. By considering ILC a morphomolecular tumor entity, it becomes apparent that ILC and NST are two entities that share a morphological overlap. And this morphological overlap is illustrated here in the schematic presentation by the dashed area. In my opinion, this morphological overlap results from at least three fundamentally different and these constellations include, first, true collision tumors, which is rare. This is an ILC growing in an independent NST. This morphological overlap includes, second, breast cancers of no specific type that mimic a lobular-like growth pattern, but they haven't lost cell adhesion. And this is common. And this is the main reason for the relative overdiagnosis of ILC in current practice. And third, this morphological overlap also includes a number of ILCs that mimic a ductal-like growth pattern. We call them ILCs with tubular elements. Now, my point is that the concepts of ILC are changing over time. And more importantly, we may even have different concepts of ILC coexisting at the same time. There is, on the one hand, the definition by the WHO, which is mainly focusing on the growth pattern of the tumor. But on the other hand, there is the concept of ILC to be a morphomolecular tumor entity, but there's also a lack of consensus guidelines or criteria to define ILC as a morphomolecular tumor entity. However, this is the reason why your research in ILC, especially clinical trials, may benefit from the review of the tumor specimens you're working with. In the last part of my talk, I would like to show that this is not just a theoretical consideration, and therefore I would like to introduce you to a very interesting tumor phenotype. We call it ILC with tubular elements. In a pathological sense of the meaning, the word elements always highlights an intratumoral variation in the differentiation of the tumor cells. I applied this term here to ILC. Just to avoid misunderstandings, I'm not going to talk about the so-called tubulolobular breast cancer. This appears to be a different story. Tubulolobular breast cancer was first described in the 70s based on HE-stained sections. A series of 4 independent papers published between 2004 and 2007 has shown that the so-called tubulolobular breast cancer is always E-cadherin positive and hasn't really lost cell adhesion. So according to these authors, breast cancer is no ILC at all. However, many experienced pathologists have remained supporters of the idea that true ILC, even if it is defective in cell adhesion, can form some tubules. It's just not clear how this works on the cellular level. And this figure shows you such an intrigue. In the region R1 of the tumor cells display a dissociated growth pattern, which is in line with the diagnosis. But in the region R2, which is less than a millimeter to region R1, the tumor cells become cohesive and form tubular elements. Judging on an HE-stained sections, pathologists might think that this is some sort of a mixed tumor or maybe a tubular-lobular breast cancer, but it's not, because immunohistochemistry for E-cadherin shows that this tumor has lost E-cadherin throughout all its tumor cells. And interestingly, this is a constellation that has never been described or investigated in detail in the scientific literature before. Now, loss of E-cadherin expression in this tumor was no surprise. It harbored the CDH1 nonsense mutation. And in line with the loss of E-cadherin expression, this tumor also showed an aberrant expression of the E-cadherin binding protein beta-catenin. Beta-catenin was lost in the dissociated growth pattern, which you can see in the left picture, but it was partially preserved in the tubular elements that you can see in the right picture. Similar findings were made for P120 catenin, which showed an aberrance in the dissociated growth pattern, but it was partially preserved in a membranous immunity pattern in the tubular elements. In Hannover, we have collected a whole series of mammary carcinomas with these features, and we've subjected them to a detailed molecular analysis. ILCs with tubular elements show a loss of the chromosomal 2, the locus of the CDH. And these tumors also harbor CDH1 mutations, mostly frameshift or nonsense mutations, and this provides an explanation for the loss of E-cadherin expression. Using very screening methods, we also succeeded in identifying the likely molecular determinant of the tubule formation. And this was actually an experiment encouraged by Patrick Derksen. So we have to thank Patrick Derksen a lot for this great idea. We discovered that the ILCs with tubular elements display a focal upregulation of an alternate cadherin named P placental cadherin. For simplicity, we call this E to P cadherin switching at the moment. Now, what's P cadherin again? From an evolutionary perspective, P cadherin arose by a duplication of the E cadherin gene. And this duplication occurred some 300 million years ago in the Carboniferous. And since then, all the higher species, including lizards, alligators, dinosaurs, birds, and the Homo sapiens, and they, in a way, harbor two copies of the E-cadherin. The one that's E-cadherin CDH1, and the other one is P-cadherin CDH3. And these genes, about 66% sequencing homology in most species. In fact, P-cadherin can function as a partial substitute for E-cadherin and can partially restore adherence junction if E-cadherin expression is gone. In the mammary gland, E-cadherin in the luminal cell layer, but P-cadherin is expressed in the basal or myoepithelial cell layer. Lobular carcinoma in situ, ELCIS, and ILC are derived from luminal cells, and that's why ELCIS is always P-cadherin negative, as you can see in the right picture. Looking at invasive breast cancers, you will find that a substantial proportion of the high-grade triple-negative breast cancers are strongly P-cadherin positive. And this reflects or correlates with a basal-like phenotype. On the other hand, P-cadherin expression is rare in luminal or hormone cancer, and it is particularly rare in ILC. In ILC, it's only about 10% that show a focal cell upregulation of P-cadherin, and those are the tumors that tend to develop focal tubular elements or compact trabecules. Now, I have to admit, I'm just a histopathologist. My profession is looking at tumor diagnostics. But by using all the fancy molecular genetic techniques, laser microdissection, SNP array analysis, and quantitative real-time RT-PCR, it is possible to formally show in selected cases that the tubular elements within an ILC are in fact just a different morphological manifestation of the same gene. It seems to be just altered by an upregulation of CDH3 mRNA and P-cadherin protein expression. Now, we've been working with E and P-cadherin for quite a while now, and we've observed some very interesting tumors. For instance, this patient was diagnosed with an ILC in a core needle biopsy in the year 2018. And this ILC showed a dissociated growth pattern and a mutation. But for private reasons, the patient didn't undergo resection of this tumor until the year 2020. When the tumor was finally resected in 2020, we were very much surprised because although the tumor tissue was completely embedded, we didn't find the dissociated growth pattern anymore, but the tumor consisted completely of tubules. Surprisingly, this tumor still harbored the same CDH1 nonsense mutation. And again, a detailed immunohistochemical workup showed that this morphological drift towards tubular elements or tubules seemed to be related to an upregulation. Now, well, for histomorphologists, it's sometimes just one or two specimens that can completely change your mind. And this tumor was such a mind changer to us. This tumor, in our opinion, implies that the E to P-cadherin switching might actually be a dynamic process. It might come and go, and it may provide ILC with a substantial morphological plasticity. Now, this has been the last slide of my presentation, and I would like to summarize. My key points for today are the concepts of ILC are slightly changing over time. ILC is still widely defined as a growth pattern, but at the same time, ILC is increasingly perceived as a morphomolecular tumor entity. And this is the reason why your research in ILC, in particular clinical trials, would benefit from a central pathology review. I believe that there's a substantial morphological overlap between ILC and NST, and E to P-cadherin switching appears to be a molecular mechanism that explains parts of the morphological plasticity in ILC. I would like to thank the clinicians of the West German Study Group, WSG, and I would like to thank the members of the European Lobular Breast Cancer Consortium. Especially, I would like to thank the pathologists organized in the European Lobular Breast Cancer Consortium. They have been working together with me on this very interesting topic of ILC concepts. In particular, I would like to thank Giuseppe Cerni, Giuseppe Flores, Gabor Cerni, Katharina Marchio, Hans Kreipe, and Anne Vincent Salomon. I would like to thank you for your attention. Thank you very much.
Great. Thank you very much, Professor Christgen. That was absolutely fantastic. I don't think I see a question in the chat box right now. And I think given that we're running a little behind, I think we'll just move on to the next speaker. So again, thank you very much. And if there are questions coming in later on, I will send them to you. So the next speaker is Dr. Peter Simpson. Dr. Peter Simpson is Associate Professor at the University of Queensland in Australia and Group Leader of the Cancer Genomics Group. I should say that we really appreciate having him here because it's like 12:30. It's like basically early morning for him in Australia. So thanks very much, Peter, for doing this. Much appreciated. The presentation of the title of his presentation is Developing LobSig, a signature capturing the molecular heterogeneity of ILC. Professor Simpson, please. And actually, I should say to all speakers, just make sure you only have one browser open. I think there seems to be interference if you have more than one browser open. So yeah, Peter, all yours. Thank you.
Thank you very much, Steffi. Thank you very much for the invitation. I'm very pleased to be with you, despite the time of being, yes, it's just gone past midnight for me here in Australia, and none of my colleagues have stayed up to support me, which is very disappointing. But yes, it's a pleasure to be with you, and I'm going to be talking about LobSig today for my presentation. So LobSig's a gene signature.
Yeah, I'm sorry, very quickly. You are not in presentation mode yet.
Oh, I'm not? No, sorry. Is that better?
No, try again now. I don't see your slides now. I think it's best if you put it in presentation mode and then share.
Yeah. Let's try that. Is that better?
No, I don't see the slides.
You don't? Okay.
Can you open it on your screen in presentation mode and then share?
I've got too many screens, which causes the problem.
Pete, if you go to PowerPoint and disable the speaker view, then you can do it easier.
How do I do that, George?
You go to the tab slideshow, and then there's you just have to remove here the presentation mode. Sorry, the presenter view. It depends on the version of PowerPoint that you're running.
Yeah. What can you see at the moment?
Nothing.
Nothing. Okay. Sorry about this. Can you see my PowerPoint now?
Perfect.
Excellent.
Thank you. That's perfect.
Sorry about that. Okay, let's go. Christine's already introduced LobSig to you in her fantastic talk earlier on. And I'll be talking a little bit about how we developed this signature and how we think it captures some of the heterogeneity that actually Christina's already talked about in the genomic context of ILC. So this is my introductory slide, and we don't really need to talk about many things on here because you're very familiar with the points that that are on the display. The key thing I want to talk to you about is that we perceive lobular carcinomas as quite a homogeneous tumor type. We talk about most of them being histological grade 2, most of them being ER/PR positive, HER2 negative, most of them being this luminal A molecular subtype. And so most of the patients have a good prognostic phenotype when we look at the histological and molecular features of the tumor. But if we consider the long-term outcome for patients, and you can see there's some survival curves over here, that the long-term outcome for patients with lobular carcinoma isn't so good when we consider the period beyond 10 years. In fact, the outcome for patients is worse than it is for invasive ductal carcinoma patients. And the reason we think this long-term poor outcome is for these patients is to do with heterogeneity. So most patients have this nice, good prognostic phenotype, and a small population of patients have unusual characteristics. We could say perhaps they're ER negative or HER2 positive or have P53 mutations. But there's heterogeneity at all aspects of lobular carcinoma, and we'll hear about the imaging characteristics of ILC later in this session. We published this a paper in 2013 showing the imaging characteristics of a cohort of lobular carcinomas, and we've since collected the survival data for these patients. And it's interesting to see that even the imaging characteristics, whether it's mammography or ultrasound, predicts a different outcome for the patients. We've just heard from Matthias, a very nice presentation about the histomorphology of lobular carcinomas, and those different types. There are a number of different variants, and collectively the variants, the morphological variants, have a worse outcome than the classic type. Now, we've also done a study recently where we've tried to do a meta-analysis of some of the large genomic studies that Christine spoke about. So we've managed to pull these data sets together to look at some of the genetic characteristics of ILC. And with a large cohort like this, we were able to look at some interesting characteristics and really confirming from other studies that things like the ER status, P53 mutation, and ERB2 mutation characteristics, whether they're amplifications or mutations, confer a poor outcome compared to ER positive tumors or P53 wild type tumors or HER2 wild type tumors. We can also see that the mutation burden may confer a poor outcome if the number of mutations in the tumor is high. So if we look at this chart at the top, we've got these three different studies. So the Euro study is from Christine, and we have the Metabrick and the TCGA studies, and we have just basically plotting the number of mutations per megabase that were found in the tumor. And most tumors have only a small number of mutations, but there's a tail in each of the studies where there's a small number of tumors that have quite a high number of mutations. In particular, here we have a tumor with a really high mutation per megabase characteristic. And we can see that this tumor is a grade 2 tumor and a luminal A phenotype. So the molecular and histological characteristics of this tumor is very standard, very a good prognostic kind of feature, but it has a really high mutation burden. We also pulled together some whole genome sequencing data for lobular carcinomas, and many of the tumors from this meta-analysis were from the ICGC, but there were two other cohorts that were analyzed as well. And I'm just showing you 4 tumors in this chart here, and the circus plot for these tumors. So if you're not familiar with the circus plots, we have the genome is organized around the outside. So we have chromosome 1, 2, 3, all the way around to X and Y. Just inside the chromosomes, we have these colored dots, and these are the mutations that were identified in tumor. Here we have green and red bars, which denote copy number changes, whether they're gains or deletions. And we have lines within the circus plot, which characterize structural rearrangements, and whether they're structural rearrangements within a chromosome or rearrangements between chromosomes. And through this analysis, we have basically, we observed that there are perhaps 4 characteristic genome landscapes, if you like, for lobular carcinomas. And this is the most common genome pattern with a fairly low number of mutations. We have a small number of copy number changes, including 1q gain and 16q loss, which Christina and Matthias spoke about earlier, and not much else going on. So quite a quiet genome. Let's put these labels on as well. There's another characteristic feature here where there's a higher number of mutations in the genome and something that we call a complex rearrangement between different chromosomes. So there's a complex rearrangement between chromosomes, and these are associated with high-level amplifications of oncogenes. There's another characteristic feature down here where you can see there's lots of mutations in the genome and lots of rearrangements and lots of deletions. And these are characteristic features of tumors from BRCA1 and BRCA2 mutation carriers. And we can call this an HR, homologous recombination deficient tumor. This characteristic is very important because these patients are likely candidates for PARP inhibitor therapy. And then the final characteristic here, which is perhaps what we're looking at the end of the tail here with a really high mutation burden, that there's thousands and thousands of mutations in this tumor, very little else going on in terms of copy number profiles or structural rearrangements. And we can call this a hypermutator phenotype and with a stable genome. So there's heterogeneity at the genomic level as well as morphological and clinical levels. And although we didn't set out to develop a prognostic gene signature or a gene signature at all, we feel like that LobSig can capture some of this heterogeneity. I'm not, in the interest of time, I'm not going to go through the method of development because we've published this, but it was based on integrating gene expression data and DNA copy number data. And the idea really was to try and understand the biology of these tumors in a bit more detail, but it morphed into a signature which we think is quite interesting related to prognosis. Like I say, we've published this a couple of years ago. What we found when we did various bioinformatic processes was a signature of 194 genes, which collectively was prognostic in lobular carcinoma patients. And you can see here in this data set of 296 lobular carcinomas, we have a signature, and when we make a score for every tumor, we can split the tumors into high scores and low and or intermediate scores, and these scores are quite prognostic for patients. The real problem for lobular carcinomas is in the grade 2 category. For those tumors that are grade 3, it's quite clear that they would likely have a poor outcome. But for the grade 2 category, this is a much more challenging, and it's very difficult to looking down the microscope to actually know whether this is a good prognostic grade 2 or a poor prognostic grade 2 tumor. But with the stratification provided by LobSig, we can see really nice stratification into good outcome and poor outcome patient cohorts. Just bring in all these annotations to show you that when we compare in an in silico analysis LobSig compared to these other gene signatures that Christine has already spoken about, we see that LobSig performs better than these published signatures. And Christine also mentioned that we think that this is probably due to the fact that we developed this signature using data derived from lobular carcinomas, whereas all these other signatures were derived from breast cancer cohorts and most likely from invasive ductal carcinomas. So we're very excited about this potential for LobSig being a predictive or prognostic gene signature. We explored the clinical relevance of LobSig by looking in tumors where we could score the Nottingham Prognostic Index. So this is a clinical algorithm using tumor size, grade, and lymph node status, and it can also include ER, PR, HER2, and Ki67. and it stratifies tumours into high risk, low risk, and moderate risk. And it's a very useful index, and it's very prognostic because it uses these characteristics that can be obtained clinically without any molecular data. You'll see from the plot here that most grade 2 lobular carcinomas are either low risk or moderate risk. When we take the moderate risk tumours, so these are grade 2 Nottingham Prognostic Index moderate score, and we overlay the LobSig molecular stratification, we get an even nicer split in the curves between low scores and high scores, which again was very exciting. It shows us that there's potential clinical relevance for LobSig in this setting, which is a very challenging clinical setting. Because all the data, all the cases that we're analyzing are from Metabric and TCGA. They all have mutation data as well, and so we can look at the mutation frequency of genes in high tumors and low tumors. And we can see there's a plethora of different gene mutations that are more common in LobSig high tumors compared to low scoring tumors. For example, P53, ERB2, NF1, ERB3, ROS1, all genes that have been spoken about already by Christine as being important in endocrine therapy resistance. So that data is all published, and so I'm going to show you some further work that we've been doing with LobSig in the last couple of years. Clearly, an in silico analysis has limitations, and the cohort size is small, and we had to use the cohort to develop the signature and validate the signature. So there's a lot of work that we need to do to show that the data is robust and worthy of follow-up. And so we've designed a nanostring assay for the 194 genes, and unfortunately we're not yet ready. We've extracted RNA, we've designed the assay, but we haven't yet performed this experiment, which is a shame for this seminar. We've also done some individual biomarker analysis using immunohistochemistry, and for this we've condensed the list of LobSig genes into a LobSig 14 gene set, which also performs pretty well on the same data set. So that a smaller number of genes is much more manageable to consider for immunohistochemistry, and we've been working through a number of these genes on tissue microarrays in lobular carcinomas with long-term follow-up. I'm going to talk to you about a couple of these markers that are worth talking to you about. So the first is NVL. Many of these genes, I should say, are quite interesting, and not much is known about them. So there's not much to go on really in terms of the literature in breast cancer or in any cancer types for that matter. But NVL was an interesting gene because it's found on chromosome 1q, and so we considered that it could be important in the amplicon that occurs on 1q. And from an RNA analysis, you can see high levels of this gene are strongly prognostic in this data set. When we optimized the antibody and we performed immunohistochemistry on tissue microarrays, this is the strongest positive case that we could find. I think there was only 2 tumors that were positive in a set of perhaps 150 cases. And so, and this was the strongest positivity we could observe. So it doesn't make for a valuable immunohistochemical biomarker for lobular carcinomas because of its infrequency in expression and the low levels of expression that we're seeing. So these are some of the challenges that we face with converting RNA data to protein data. The second marker is an armadillo repeat containing X-linked protein 2. It's got an interesting name. Again, not an awful lot is known about this protein, but the antibody works very well. You can see the speckly brown staining around the nuclei of the tumor cells here. So this is a stromal marker actually marking mitochondria, according to the small amount of literature that's out there for this protein. So we have quite strong staining in the cytoplasm. So we've scored a lot of these markers based on intensity of expression and cellular localization. And there's a few plots here to look at if you're interested. I'm just going to go straight to this plot in terms of looking at whether the intensity of expression of this protein is prognostic. So we have 4 categories of staining: negative, a kind of a weak, wishy-washy, positive staining, and some more robust positive staining, which has been grouped together. And you can see the high levels of expression are predictive of a poor outcome in this data set. The next candidate gene is this SLC44A1, which, and I've got 2 examples here of 2 tumors. This one is negative, but you can actually see nice staining in the blood vessels. So it's actually a nice internal control for the antibody, working quite well. And then there's another staining where the tumor cells are staining positive. This kind of characteristic is going to be a confounding factor in the RNA analysis for tumors that are highly vascular. So it might look like the expression of this RNA is quite high in the tumor, but it would actually be coming from the blood vessels. Again, there's a couple of charts here for you to look at if you want, but we'll just go straight to this image here. And we can see that positive staining for this marker, again, is prognostic in this data set. The final marker I'm going to show you is this TSHZ1, which seems to be a DNA binding protein that regulates transcription, particularly in development. And again, I've got 2 tumors to show you. This tumor, we can see brown staining in the nuclei, and so that this staining pattern is nuclear only. Again, we graded the staining as in strong, moderate, and weak. And this tumor, hopefully you can see the nuclei are sometimes blue and sometimes brown, and the cytoplasm is brown. So this is telling us that there's cytoplasmic and sometimes nuclear staining in this category. So we've stratified the cohort whether they were nuclear only and nuclear and cytoplasmic staining. So the grades of expression of this protein had no impact on prognosis, but the cellular localization did. So when the protein seems to be able to shuttle between the cytoplasm and the nucleus, it predicted a worse outcome in this data set. So there's some interesting things to think about with all of these proteins, different characteristics in terms of the scoring, and what characteristics might be of interest biologically. We're really interested in combining markers in the same way that we've combined messenger RNA for different genes into a signature. And so just showing you the combination of these 2 proteins, and we've seen these 2 charts already. Both of them are predicting poor outcome. If we just take up this group of patients that are strongly positive for ARMCX2, and we overlay SLC44A1, which is either positive or negative, we can see the addition of SLC44A is able to stratify this line into a good group and a worse group. Likewise, if we take this poor outcome group of positive staining SLC44A1 tumors and stratify by ARMCX2 into low and high, we can see the same thing. A positive group and a positive negative group. So that combination of markers is able to enhance our capacity to pick up poor outcome tumors. So I think combining markers is going to be an interesting way to go when we've developed more markers and have more data. So the goals for LobSig in the we're just working through this project. We've developed another tissue microarray to increase the numbers of tumors. Like I mentioned, we've got this nanostring assay that we're hoping to work through in the next couple of months. And we're collecting all this sort of information to enable us to do a proper evaluation of individual biomarkers and the nanostring assay together with these clinical, morphological and molecular biomarkers. Particularly interested in to see whether the biomarkers or the signature as a whole can stratify better than grade or better than the mitotic score, better than KR67 staining, for example. So it could be that LobSig, either individual biomarkers or a nanostring assay, could be a companion diagnostic for lobular carcinoma patients. And we can see possibly, as in for other gene signatures, low score patients may not need might gain no additional benefit from further molecular testing or chemotherapy. But high score patients might warrant increased surveillance, for example, or comprehensive molecular testing to identify the gene mutations or the gene characteristics that could be causing this high score. And maybe they can be candidates for targeted therapy based on the molecular testing that was applied or chemotherapy approaches. So just to finish off this presentation, we have this impression that lobular carcinoma is a homogeneous tumor type. It has that classic infiltrating morphology, ER, PR positive, HER2 negative, quite indolent, and a good response to hormone therapy. But actually, there's significant heterogeneity, whether we're looking at imaging, morphology, immunophenotype, and molecular aspects, whether these are gene mutations, amplifications, or mutation, more global genomic signatures. We think LobSig has the potential to capture this heterogeneity, and it also gives us insight into the biology of these tumors with poor prognosis. I'm going to finish with some acknowledgements. There's a suite of people here from our group, past and present members of the group that have contributed to the project. Special acknowledgement to my mentor, Sunil Lakhani, and now colleague. I could have a nice photo of George here because we conducted some very nice studies on lobular carcinomas in our younger days. Special mention to Amy, Jamie, and Samir, who've really been great collaborators, great colleagues, and have contributed significantly to the work I've presented today. As with Christine, I'd like to thank the funding bodies and the tissue banks, and most of all, the patients that have provided material for us to work with in the research setting. So I'll stop there. Thank you for listening.
Thank you very much, Peter. That was great. We would have loved to see a picture from George from the earlier days. So next time, put it in. So anyway, let's see if questions come up. I think we have time for one or two questions. Maybe I can ask you a question. There's a lot of interest on LobSig in the advocacy community. I have heard a lot of discussion and question, and at the same time, there's a lot of discussion on really increasing international collaborations. And, Christine talked about this earlier. Do you think that could be the validation of LobSig could potentially be something that would be a great, base for getting various groups together and doing that?
Absolutely. I'm glad you brought that up, Steffi, because that's exactly what we need to take this to the next level. So we've been thinking about as soon as the nanostring assay has been performed and hopefully performs well, and as soon as we've got some nice biomarkers that we're really interested in pursuing, we would very much intend to reach out to people for help to take it to the next level. Absolutely.
Great. And then there's one from Christine. She's asking whether on the same tumors where you do your, the nanostring IHC, do you also intend to do other commercial tests, the regular tests on the same samples?
So we do have the possibility of doing that, yes, because we've extracted RNA. We should have enough to do that. It wasn't part of our thinking purely from a funding point of view, but we could. That would be the best thing to do, is to really compare directly on the same RNA samples.
Okay, that's great. And quickly, what was the rationale on the N14, on the IHC panel to pick those 14 genes on the smaller panel? How did you do that?
It was a statistical test, a Cox boost analysis, and that was it was a way of just eliminating perhaps genes that had some redundancy in there.
Okay, great. And then the last question is, given the heterogeneity of the disease, which we have heard, about earlier in the lectures, are you worried that the test run on the core biopsy or in one section might miss, important features of the disease in the test?
Yeah, that's a great question, and it's something that we have thought about. We would have to do quite a bit of optimization or investigation about multiple regions within a tumor, different tumor areas with different tumor cellularity and stromal composition, to see how the score varies with those different parameters. So yeah, it's something that's very important to take forward. Yeah, absolutely.
Okay, great. Thanks, Peter, again.
Thank you.